Hello everyone, welcome back to one more video session. In this session, I'll be speaking about a very important concept, that is Hershey and Chase experiment. Okay, you have to listen to me carefully. Very important. Conducted in the year 1952, and it is also the proof for DNA as the genetic material. It gave the proof that the DNA is itself genetic material in living organisms. In 1928, we know that the Griffith conducted one experiment, and ultimately he said there was something else, a type of biochemical present within the bacteria got incorporated or picked by another bacteria, which caused the transformation of that bacteria. Okay, so that is transforming principle. And after that, every McLeod and McCarty conducted biochemical characterization experiment of the transforming principle, and they proved that the DNA is itself genetic material, and even it is the transforming principle of Griffith. But the scientists, they did not believe that the DNA is the genetic material. According to them, still it was the protein as the genetic material. To disprove them, as well as to prove DNA is itself our genetic material, which is responsible for the expression of character, because in the DNA we have the genes responsible for the production of proteins, and proteins are responsible for the expression of characters in all living organism. Okay, which was given by Hershey and Chase experiment. Finally we came to know that yes, DNA is the genetic material. Okay, so very important. And here, before we go into the experiment of Hershey and Chase, we have to go back to our first year PUC to see and to learn certain basics or recall certain basics. Yes, obviously you could see here, this is the virus. I have used white color here, and the green color, this green color thing is the DNA or the genetic material. Okay, we just write DNA, genetic material, and this everything is the protein coat, the outermost layer of the virus. We call them as capsid. Capsid is nothing but protein, correct? So we know it. And if the virus causes infection in the bacteria, then that kind of viruses are called as bacteriophage or bacteriophage. Just remember it. And here, before they conduct the experiment, they grew these bacteriophages in two different types of medium. In one medium, they put a type of isotope called as sulfur 35 isotope. It is actually which will radio label only protein particle of the virus. That's what I have used the pink color. You could differentiate now. This is normal virus, normal bacteriophage. This is the normal DNA. The green color is normal. It should be understood like that. Whereas white color is normal outermost layer, the capsid. Here we have used pink color. That means because these viruses were grown in a particular type of medium containing S35, that is sulfur 35, which is a type of isotope of sulfur, which radio labels only proteins. Why it radio labels only protein? As a result, you could see the change in the color of the capsid. Now it is in pinkish in color. It is now not normal. It is radio labeled. Okay, I'll write here. It is radio labeled capsid. RLC, radio labeled capsid. What is the reason why sulfur 35 now binds to the capsid? What is the reason? Because if you look into the capsid structure, or if you look into any amino acid in the sense, or any proteins, not amino acid, protein, they have amino acids. Like there will be so many amino acids which are linked together by peptide bonds to make polypeptide chain. Particularly, majority of the proteins contains a common type of amino acid called as the methionine and cysteine. Methionine and cysteine are the common normally seen type amino acids in polypeptide chain. So even the capsid also contains methionine and cysteine. This S35 always binds to methionine and cysteine of the polypeptide chain, because these two are sulfur containing amino acids. That's what sulfur now binds to sulfur itself. That's what actually the entire protein now becomes radio labeled. That's it. This is the thing. Whereas in this case, they grew another type of the same type of bacteriophages in another medium containing another type of isotope of the phosphorus, that is P32, phosphorus 32. Okay, it is another type of isotope of the phosphorus P32, which labels or radio labels only DNA. It will not radio labels the outermost layer, or it will not radio label the capsid. It will radio label only the DNA, because In DNA, if you look into the structure of the DNA contains phosphorus. That's it. So this phosphorus now obviously binds with this phosphorus, makes the DNA radio labeled. This is it. What is this radio labeling? Radio labeling is a technology or technique used in biology to see or to trace any kind of molecular movement within a like cell. Okay, so this is the thing. So if they radio label it, obviously they could easily trace the movement of any kind of particle, any kind of biomolecule within a cell. This is actually a commonly used technology or technique in biology, radio labeling. Okay, so there are so many techniques to identify radio labeled molecules. We will not talk about it now, it's a lengthy process. Here we say that, you know, okay, so in this case we have the capsid radio labeled bacteriophage, and in this case we have DNA radio labeled bacteriophage, correct? Okay, so we write here P32 and in this case S35. Just remember it, sulfur will only radio label the outermost layer, whereas phosphorus only radio label the DNA. That's what, like I have used white color normal, whereas DNA is in bluish color because it is now radio labeled by the phosphorus 32. In this case pink color, it is radio labeled by S35. This is it, simple. Whereas the DNA remains as it is, that is greenish in color, that is normal. Okay, now they started the experiment. Here they have used a type of host which is Escherichia coli or E. coli. Simply we call it as E. coli. Okay. Now we have to learn what is exactly the kind of life cycle exhibited by these bacteriophages. Both the bacteriophages are same. They belong to the same species. Okay. So here what we do is I'll just write two types of life cycles. One is a common type of life cycle exhibited by these bacteriophages called as lytic life cycle. The other one is lysogenic. Even though this is not necessary for our experiment, but you need to know the basics. Lytic means whenever the bacteriophage actually causes the infection. You know what is infection? Infection means, yes, a bacteriophage sits on the membrane or cell wall of the bacteria, then actually it inserts its genetic material. In the bacteriophage, the genetic material is DNA. There are viruses with double stranded DNA, single stranded DNA, or single stranded RNA, double stranded RNA. There are so many. Okay, it is classified by Baltimore, like class 1 to class 6. You are not going to study it in PUC level. You will, if you take medical field, obviously you are going to study about Baltimore's viral classification. Okay, and here you could see that, you know, this is the DNA. It gets inserted within the host. Host is E. coli. This is it. This is infection. Once the DNA of this virus or the bacteriophage enters into the host cell, they get incorporated within the genetic material of the bacteria and it hijacks the bacteria, and now the bacterial DNA acts as if they are the viral DNA and they starts producing the viral particles. This is a type of life cycle exhibited. This is common for both lytic as well as lysogenic till there. And after that, if for example, if this is the bacteria and ultimately so many bacteriophages are produced, if the number increases, obviously it causes certain pressure within the bacteria and leads to the rupturing up of the bacterial cell wall. That's it. That means the death of the bacteria. That is lytic, and it is very, very fast. It's very, very fast process. Whereas lysogenic is not like that. HIV is a very good example. Like, you know, it normally resides within a cell and then actually it produces its viral particles, but it will not cause the rupture of the cell. Like it takes a lot of time. It takes around 2 to 8 years. Whatever that, always remember that there is always one incubation period. Incubation period in the sense like it is the time taken by the virus to produce the viral particles. That's it. Okay. In this case, incubation period is very short. In this case, incubation period of the bacteriophage is slow. It's very, very slow. That's it. This is the thing. Here, this type of bacteriophage exhibited lytic, that is very, very fast. Okay, lytic life cycle. That means it will cause the rupturing up of the E. coli. It will cause the death of the E. coli. This is the thing. Okay, you will understand why they have used or preferred this type of bacteriophage which exhibited lytic life cycle. Let us see that. This experiment is understood by three steps. The first step is infection. You know what is infection? Okay, blending and centrifugation. Very simple. Take two Petri plates. In one Petri plate, just put some E. coli, Escherichia coli, around like let us say 100 E. coli, and in another Petri plate you put around another 100 E. coli. That's it. That's the same thing they did. And after that, for one Petri plate, they actually put some this type of bacteriophage which is already radiolabeled by the S35, that is radiolabeled protein bacteriophage. You could see here I have used pink color, it's understood. Whereas to the another Petri plate containing E. coli, they poured the radiolabeled DNA containing bacteriophages. That's it, very simple. Okay, and now this will lead to infection. You know what is infection? That is the this particular bacteriophage, both the bacteriophage will insert their DNA. In this case, they will insert normal DNA. In this case, they will insert radio-labeled DNA. That's it, P32 labeled DNA. And after that, you know that this cannot enter into the cell of the E. coli because the protein will not enter, the capsid will not enter, only the genetic material will be enter. That means the virus is sending its message to produce its particle. That's it. It's a kind of they sacrifice their life. But obviously the message is sent into the host cell to produce numerous or many number of virus inside and causing the death of the E. coli. Okay, that's the thing happened. And I have used a red color here. This red color one is the genetic material of the bacteria or E. coli, and this green color one is the genetic material of the bacteriophage which is not radiolabeled, and obviously they will incorporate and they merge, and as a result it starts producing lot of particles, that is viral particles. Even the same thing will be applicable to this, but in this case it is radiolabeled. Okay, so ultimately many number of viral particles will be produced. The second step is blending. Once the viral particles are produced, even though in the diagram I have not mentioned it, but in the third diagram I have mentioned the production of viral particle doesn't matter. Actually, it is understood. This is what is happening within the E. coli, what is happening within the or inside the cytoplasm of the E. coli. This is experiment. Second step of the experiment is after they allowed it for some time and they knew that obviously the viral particles are now produced within the E. coli. Now they used another method which is called as blending. Blending in the sense like shaking. They shook the entire Petri plate, okay, using a blender, a machine. Okay, it is not your mixer grinder, it's entirely different. It is used in biology field. It's normally used to shake the cells of like tiny cells. Obviously cells means it is tiny itself. Okay, so like that they used it due to blending or due to shaking. Obviously if the bacteria like cell wall is almost is about to rupture, it will rupture. Correct, it is going to rupture in the sense like whatever the like particles here we just see here, whatever the parts which are there inside this bacteria will come out. That's it. This is the thing. Okay, so because it's about to rupture, because of the shaking, it will get ruptured, and whatever the particles which are present, particle in the sense viral particles, will come out within the solution. So in the Petri plate, you could see a solution itself, solution of bacteria particle as well as the viral particles, both will be there. The same thing is applicable here also. It is understood like that. After that, centrifugation. Everything like whatever there in the Petri plate, two Petri plates into a test tube. That's it. And then centrifugation is nothing but it's like spinning. Obviously test tube means it contains the solution where it contains bacterial particles as well as the viral particles, and due to spinning based on the gradient method, you get two fractions in the test tube. If you look into the test tube, you get two fractions. So I will just write here I'm drawing one test tube for this experiment, and I'll draw one more test tube for this experiment or this particular solution. What you get is, okay, so we just use this color and white color. This white color is the solution. Solution is always supernatant. You know about supernatant, okay, and this one is the pellet. Pellet is the solid, that is it contains virus as well as bacterial particles. It is solid, normally settles at the bottom of the test tube, whereas supernatant it will be liquid itself. And in the here also same case, supernatant and the pellet They got the same result in both the experiment. Okay, so this is the pellet. And after that, after the centrifugation, now they checked what is there in the pellet of this particular experiment. That is, I'm talking about S35 radio labeled protein bacteriophage. Here they did not find anything, nothing. Even though DNA was there, normal DNA of the virus was there, but it was not radio labeled. So obviously you cannot see it in any machine. Okay, so they did not say anything else. Here they found S35, that is radio labeled capsids floating. Please remember this, S35, that is radio labeled capsid of the bacteriophage, was found in the supernatant. This is very important. It is 100% sure that the entire, the capsid did not enter into the bacteria. This is very, very important. If it has taken the entry, in the sense like if the protein taken the entry into the bacteria, obviously you could see them, you should see them in this pellet, but obviously the protein particle did not enter into the bacteria, and obviously it was floating in the supernatant. This is the one thing. Whereas in this case they did not find anything else, because in this case they did not radio label the capsid. Obviously they could not see anything else because there was no machine to see the protein as it is. But in the pellet they saw the DNA particles of the bacteriophage because it was radio labeled. Now it was sure that the DNA took the entry into the bacteria, whereas the capsid did not take the entry. So this DNA now caused the lysis of the bacteria, killed the bacteria. That's it. That means the genetic material, that is the DNA of the virus, entered into the bacteria. That means it is 100% sure that the bacteria is itself the genetic material which caused the production of more viral particles, as well as it caused the death of the bacteria by causing lysis. That's it. So it proved it. This is what actually you need to understand. So where exactly they found the DNA, that is radio labeled DNA, in the pellet particle, that is the solid particle of the solution, whereas S35 was found in the supernatant. That's it, because it is understood if S35 was present in here the pellet, obviously that means the protein was the genetic material, but no. In this case, the DNA entered into the bacteria and caused the death of the bacteria, and they they recovered the radio labeled DNA in the pellet. That's it. So it is understood that DNA is the genetic material of the living organism, even it is in virus or maybe in the bacteria. That's it. So this is actually the thing we have to understand. So that's all about Hershey and Chase experiment. I did not write their full name, Alfred Hershey and Martha Chase. Just remember it, very, very important. It could be asked for your board as well as NEET. Hope you understood this concept. Very simple. For notes, you have to refer the class notes given by me. Very simple. Okay. So no need to, you have to draw the diagram of all these things which is already given in the notes. Okay. So fine. Explanation part is also very easy.